In this short video, we're going to be talking about RNA. RNA is a distinctly unique type of nucleic acid in its ability to have simple linear forms, but also to form complex secondary structures or complexes with proteins or other molecules. RNA is transcribed, meaning it is copied from a DNA template. However, when RNA is created, we replace T, thymine, with U, uracil. RNA is synthesized as a single strand. Most commonly, when we think of RNA and transcription, we think of mRNA. mRNA comes in two flavors: pre-mRNA and mature mRNA, which we will just call mRNA. Pre-mRNA can also be called a primary transcript, and is the first thing that we transcribe in eukaryotic cells. That's because mRNA in eukaryotic cells is going to be processed inside of the nucleus before it enters into the cytosol. However, in prokaryotic cells, mRNA can be synthesized or transcribed and translated at the same time. Mature mRNA, whether in prokaryotic or eukaryotic cells, is what is translated. This means the information stored in a strand of mRNA is used to create a protein. The next type of RNA that we'll look at is tRNA. tRNA can be represented as a flat cloverleaf with 4 distinct arms and 1 accessory arm. Let's start with the first arm, the acceptor arm. The acceptor arm contains a highly conserved sequence called the CCA sequence from 5' to 3'. Now, if any of y'all fish, you can think about this like the Coastal Conservation Association. So remember this, whenever you think of the acceptor arm, think about fishing. So there's a 3' end with the A at the end of it. And we'll look at why this A is important in a minute when we look at tRNA charging. But now let's look at the T arm. The T arm contains a specific and special rare base called Psi, or pseudouridine. Pseudouridine is just a modified form of uracil, but that is a specific residue that you'll find on this arm. The next arm is the anticodon arm. The anticodon arm is very important. So if the tRNA is facing like this, essentially our anticodon is going from 3' to 5'. Thus, this tRNA will be complementary to an mRNA sequence with the 5' to 3' sequence UAC. Finally, we have the D arm, or the dihydrouridine arm. Dihydrouridine is another modified form of uracil that we find on this particular arm. And although we often think of tRNA as this flat cloverleaf structure, in reality, tRNA's structure follows a lot more closely to an upside-down L shape with a 3' end, which is often charged with an amino acid, which we can call an AA. In a later video, when we're looking at translation, we'll actually look at the mechanism by which the tRNA is charged. Next, we're going to look at 2 types of RNA together, and we're going to look in a specific part of the cell and a specific part of the nucleus called the nucleolus. The nucleolus is highly important to the formation of rRNA through the action of snRNA. snRNA, remember, stands for small nucleolar RNA. And what happens is that synthesized rRNA, or ribosomal RNA, is folded in the nucleolus by snRNA. Like with other types of RNA, rRNA is formed as a strand of RNA. And in the nucleolus, what snRNA is able to accomplish is folding this rRNA into complex structures known as the small and the large ribosomal subunit. These ribosomal subunits will be transported into the rough endoplasmic reticulum as well as into the cytosol. Ribosomes serve as a location for translation. Importantly, the large subunit of the ribosome contains a particularly interesting property, peptidyltransferase activity. Peptidyltransferase ability means that the large subunit of the ribosome has the ability to catalyze the bond formation of a peptide bond during the synthesis of a protein in translation. Let's now look at snRNA. snRNA is a special type of RNA that will process pre-mRNA into mature mRNA. We can think of snRNA as a simple formula. A molecule of snRNA plus proteins will give us an snRNP. Remember from the telomerase video that RNP means a ribonuclear protein. And if we have 5 snRNPs, we have a spliceosome. A spliceosome is the core unit of snRNA's activity. snRNA will cut out introns, sections of RNA that do not leave the nucleus, and ligate back together exons, the section of RNA that go on to be translated into a protein. Although in this class we primarily talk about mRNA processing, there's other types of processing such as tRNA processing, or as we described before, rRNA processing. However, again, for the purpose of this class, when we're thinking about snRNA and forming spliceosomes, we're talking about that in the context of processing pre-mRNA to mature mRNA that can be translated in eukaryotic cells. Next, let's talk about RNA interference, or RNAi. RNAi is analogous to an immune system for our genome. Let's first look at siRNA. siRNA is formed in response to exogenous sources, so outside sources of double-stranded RNA. These double-stranded segments of RNA will be chopped up by a protein called dicer. Dicer will make small segments of RNA, which we know to be the precursors for RNAi. Each of these strands can be separated and, when combined with a protein, form a complex known as a RISC, or RISC, which stands for RNA-induced silencing complex. This RISC, which is associated with an siRNA, can perfectly bind to a target piece of RNA, like either strand from before, and will cause endolytic cleavage and degradation of this RNA, preventing it from causing infection or damage to the cell. Next, let's look at microRNA, or miRNA. miRNA differs from siRNA in that the source of miRNA is endogenous, meaning that the cell is creating it. A small molecule of RNA is transcribed with a hairpin loop in it. This molecule of RNA is called a priRNA, or a primary microRNA. This primary microRNA undergoes cleavage in which these outside portions are removed, leaving only the hairpin. Finally, these small segments of RNA are associated with a protein, and like in the creation of siRNA, they form a RISC complex. Now this RISC complex, unlike siRNA, will not bind as tightly, and thus, instead of catalyzing cleavage, it actually inhibits translation by blocking a sequence of RNA from being able to fit through translational machinery. Because miRNA binds less tightly than siRNA, it has a wider specificity, and miRNA can recognize about 60% of the human genome, giving the ability to control and regulate translation. The CRISPR-Cas system is highly similar to RNA interference, except CRISPR-Cas only occurs in bacteria. There's 2 phases of it, acquisition and expression. Let's first look at acquisition. During acquisition, DNA from a phage is processed and inserted into a portion of the bacterial DNA called the CRISPR array, where other pieces of phage DNA have already been incorporated. Each region of DNA acquired from a phage is called a spacer, and in between each of these spacers is a palindromic segment of DNA that's recognized by restriction sequences that allow the DNA to be opened up to insert these spacers. Next, let's look at the expression of CRISPR-RNA. In expression, we begin by transcribing the CRISPR array into a strand of RNA. Cas proteins, which are proteins important to the CRISPR-Cas system, process this RNA, and in the case of the pink one, we'll just say this is the strand that we've processed and that our Cas protein has associated with. So after the Cas protein associates with it, we have an effector complex. The RNA in the effector complex is complementary to the DNA in the phage. Thus, if this phage DNA ever presents again in this bacterial cell, the effector complex may bind to that DNA and cleave it. Let's take a look at that. Let's now look at inhibition. So let's imagine that the phage from earlier presents its DNA in the same bacterial cell again. Let's look at what happens. The effector complex will bind to the DNA strand. It will split it open using helicase activity, and the RNA sequence within the effector complex will complementarily bind to that DNA. The effector complex will cleave the infecting DNA and then dissociate, thus leaving the phage infection inactive. I hope you found this video really helpful. The concepts and information presented in these videos will be true no matter what genetics class you are taking. However, the concepts presented in this video are referencing material currently covered in Baylor University's coursework. Remember, if you are currently enrolled as a Baylor student, we offer free tutoring services. Our tutoring center is located on the first floor of the Sid Richardson building. You will find all the details you need to know about these services on our website, www.baylor.edu/tutoring. You may schedule a free 30-minute one-on-one tutoring session online through Navigate, or just drop in during our open business hours. For more information about our current services, please visit our website. Thank you.